Good morning, everyone. We welcome you back to the second part of session 1. Okay. This session will be chaired by Padma Bhushan awardee, Professor G. Padmanaban, who needs no introduction to this gathering, a PhD graduate from the Department of Biochemistry and former director of IISc. Professor Padmanaban paved the way for recombinant DNA technology in India. He discovered the heme biosynthetic pathway as a target of drug therapies against malarial parasite and also established the efficacy of curcumin-based combinatorial therapy for the treatment of malaria. He has been part of several government agencies and chaired multifarious committees. Professor Padmanaban has been the recipient of many distinguished awards, including S.S. Bhatnagar Prize for Science and Technology, C.V. Raman Research Professor, and Jawaharlal Nehru Bird Centenary Fellowship of INSA, and many more. Without further ado, we'd like to invite Professor Padmanaban on stage to introduce Dr. RENU SWAROOP.
Professor ANURAG KUMAR, Director, Institute of Science, Dr. RENU SWAROOP, Secretary, Department of Biotechnology, Professor RANGARAJAN, Chairman, Biochemistry, Professor D.N. Rao, convener of the whole celebration, many distinguished colleagues, students. I have great pleasure in introducing Dr. RENU SWAROOP, Secretary, Department of Biotechnology. I have been connected with the Department of Biotechnology even before it became a formal department in 1986. It was started as National Biotechnology Board in 1982. So I have very great pleasure in introducing her. I'm going to take full 5 minutes. DBT has been a major benefactor of this department, as the division, as well as the institute as a whole. All the faculty would know her, but a formal introduction is needed on an occasion like this, and let all students know, as well as guests know her background. Dr. SWAROOP has a PhD in genetics and plant breeding and a postdoctoral stint at John Innes Centre, Norwich, UK. She chose to become a science administrator. Her record as science administrator at DBT for 28 years made her an automatic choice to become secretary of the department for the last 2 years. She has contributed significantly for the growth of life science research in this country. But I will confine to aspects of biotechnology. And there are many examples. I don't have that much time. The unique example is the establishment of BIRAC, Biotechnology Industry Research Assistance Council, as a Section 8 company of DBT. It has become a flagship institution in the country. Apart from the fact that SWAROOP is the author of the process of consultation, EFC cabinet approval, registration, and incorporation of the company within a short period. You know, to establish a company, that too within a government system, is not easy. It has given rise to 2,000 startups in the country, 50 incubators. And the plan is to have 10,000 startups and 250 incubators. 130 commercial products have become a reality. She played a major role in setting up the National Biopharma Mission with 750 crores from the government and 750 crores from the World Bank. The objective is to make specific biopharmaceuticals, devices, and diagnostics within a period of 5 years. And 3 years are left. The government has put a target of $100 billion for biotechnology industry by 2025. Dr. SWAROOP has catalyzed the initiation of mega projects like reference genome of the Indian, with about 20,000 Indians from all over the country, which still is a small number compared to the population, but we have to start somewhere. Also initiated participation International Bioearth Genome Initiative, where India is planning to sequence 500 animals and 500 plants unique to Indian biodiversity. Catalyze the gut microbiome project, and objective is to make at least 6 vaccines, and I believe even corona vaccine is included there. You all know probably India developed the first indigenous rotavirus vaccine, and years ago this department developed the recombinant hepatitis B vaccine. And most important is also her initiative to start biological data policy and biological data center. Many of you who are submitting papers would know your sequence or image data has to be submitted to EMBL or to NCBI. We don't have a biological data center in this country. And she has taken this initiative. It's not an easy exercise, it's a huge exercise. Similarly, many contributions of her have been made in the area of agriculture, whether it is secondary agriculture, whether it is soil health card issuing. No, these days soil health has become very important. Providing breeding materials and so on. And there are many other examples of her involvement in policy. Recently, she served as the convener of the technology group constituted by the Honorable Prime Minister to coordinate and present the vision and roadmap for 11 ministries and under technology sector. And she has been coordinating with all the ministries where MOUs have been signed, ICMR, ICAR, CSIR, DAE, Ayush. You know, MOUs normally tend to be on paper. And she has tried to give meaning to all these interactions with other ministries. At the international level, there are many examples. Setting up of the India component for the Coalition for Epidemic Preparedness for Infections, Global Antimicrobial Resistance R&D Hub with 16 countries, Mission Innovation for Clean Energy with 25 countries. At present, she is the vice chair of this steering committee. Indo-UK Crop Science Fellowship Program, Business Finland, and many more. I just try to give a glimpse of what she has done and what she is capable of. She is a dynamic administrator, and there are many initiatives taken by her to revamp DBT itself. We all have complaints now. At the same time, we saw totally dependent on this department. And motivate performance internally, motivate and help. There are 13 or more DBT institutes all over the country who have been doing basic research like us, pushing them to do translation. And she has interacted with the scientific community and the industry at large. She is a recipient of several awards. I will not list them. The only one I will mention is she is the recipient of the National Entrepreneurship Award 2016-17, even before she became secretary. The most prestigious award of the government, which she received from Mr. Arun Jaitley, the late finance minister. Dr. SWARUP, I request you to give the address to this audience.
Thank you very much, sir, for this very, very kind introduction. Makes me feel truly humbled. But I think coming to these centenary celebrations for the Department of Biochemistry itself is a huge honor, I would say. And thank you, RANGARAJAN, and all of you for inviting me. Because this, as you've said, the IISc as a whole, but specifically this department and its engagement with the Department of Biotechnology goes back to since when the Department of Biotechnology was born. And as Professor Padmanaban said, even before it became a department as a board, since this is 100 years and DBT itself is 34 years, so we can clearly say that. And I do recall that when I joined the Department of Biotechnology in 1989, if I remember correctly, the first task force committee as a junior scientist that I sat and we did the recommendations, and if I remember correctly even now, I think it was Dr. Swaminathan who was the chair of that task force, was a project which had come from this very department that we had to take forward. So it goes back to so many years of association, and at this point my heartiest congratulations to each and every member here, to the department, to the institute, to the alumni, to the faculty, to the students, to the young students who are now the future of this department, and very rightly said, it's 100 not out. I totally agree with that. It's 100 and many more hundreds that we would really want to see this department moving ahead, because I think the outstanding contributions which this department has made to the whole area of life sciences is something that we all respect, and we would want to see this being carried on, because not just the research contributions which this department has made, but the numerous scientific leaders which this department has created, the numerous scientific collaborations which this department has brought out into the area of life sciences. It is very important that this department stands soundly to be able to build it as we take on. And I think the Indian Institute of Science, which is itself a name, and then having this department here, it's not just nationally, but I think across the globe there are huge collaborations which have their foundations in the research which began from here. If you look at the history of the department and in fact of the institute, I was looking at the slide which the director showed. It clearly gives you how the institute has evolved, and within that this department, the way we are looking at moving dynamically as science is moving, bringing in the emerging tools, bringing in the emerging technologies, looking at newer areas, because that's what all science is all about. How do you keep pace with the changing science, with the dynamically fast moving science? And I think that's clearly and solidly a reason why this department has sustained itself so well during these 100 years, and we hope as it moves on, because you're keeping pace with this changing dynamics of science, and I think that's very important. It's important, and this is mainly for the younger students and researchers here, to keep pace with that, so that, and keeping pace with newer technologies, newer emerging tools is definitely not easy. It pushes you out of your comfort zone at many times, and you're trying to look at newer ways, but that is the way to move ahead. If you look at all the various activities which this department has been engaged with across different disciplines within this department itself, the contributions it has made into the, as I said, of course, the scientific leaders, but into building these centers of excellence which go around team leaders, the publications, etc., looking at various aspects, whether it's new knowledge when you're looking at the, well, you have the biochemistry department, it encompasses within itself many newer disciplines and areas which can become a part of it. And I think that is really important, because this is truly a very good example of how the basic sciences moves across that value chain, from moving from basic knowledge generation, fundamental, applied, to actual translation and product driven. Professor Padmanaban just spoke about how the vaccine which was developed by this very department was the endpoint product that you talked about. And then we know of the excellent basic research and knowledge generation which has come in from a large number of publications and other research which has happened. So I think this is key on how this has been the history of this department over the last 100 years. As I said, it's not just this department as itself, but I think there have been centers which this department has been responsible for creating. And those are key and critical into this larger ecosystem of science, research, innovation that we talk about. Within this division of your biological sciences itself, you have so many departments which are so closely and intricately associated. But I think it's also the confidence that various government funding agencies have had in the strength of this institute and specifically in the context of this department to be able to invest in different research opportunities which this department has been able to take forward. And it's all the various funding agencies, whether it's the Department of Biotechnology, whether it's DST, whether it's CSIR, and I know that this department is been a recipient of many of these across the various disciplines that you're working into. But what is also very interesting, and when I walked in, you were talking about the alumni funds which are coming in. I was just talking to your director to say that it's interesting to see that now you have been able to attract a lot of philanthropic funds. And I think that is very, very important. Not just because it's a mix of government and philanthropic funds, but it gives you a different mindset, it gives you a different culture on which you start working when you are attracting funding from these various organizations. We all look at the core funding which goes into departments and to institutes, and the DBT, as Professor Padmanaban said, and most of you know, we itself have 16 of our institutes. Each of you have a core budget, but in addition to that, scientists are always encouraged not just to bring in funding, which is of course key and critical, but to go and compete for projects, to go and bring in funding from outside, from other agencies. Why? Because it helps you to get into that whole competitive spirit of being able to see where you are vis-a-vis your peer. And I think that is one of the parameters with which the performance of the institutes or the departments is judged. How much of extramural funding are you being able to bring in? Because if you are bringing in extramural funding, means you are being able to compete at that level of national and international funding, and that determines the quality of the science, and that I think is again is something which this department needs to be really proud of. I think it's also important to see that while this department has collaborated a lot with the international agencies, but we've also seen collaborations which are happening with the industry. So across all stakeholders, there are programs which you're doing with CSIR, there are programs which you're doing with AYUSH and others, and I think this indigenous priority identification and then picking up interdisciplinary areas of research, combining them together, and then being able to find a roadmap for yourself is something which is important and critical, and we'll see how that is really important to shape the future progress of your own department. As I said right in the beginning, this department has not just been associated with DBT in terms of being a recipient of funding from DBT, but I think most of the faculty, especially the senior faculty here, have been part of the DBT's own scientific due diligence process, which DBT and all government agencies, you know, rely heavily upon. All our entire scientific funding that we do, our whole planning that we do for programs, it's not just done at the center sitting here, it's a consultative process in which we engage with scientists across the country. And I can sort of look around and say each one of you has been so closely involved in this. Professor Padmanaban, of course, has been the chair of, I think, mostly every committee in DBT, and we hope, sir, you do continue. He's the chair for our scientific advisory committees, and he's the co-chair right now for our Apex board, which takes the final decisions. But each of you, I mean, I really cannot even name them because each of you has been engaged in different components of it. So that goes to say how deeply engaged you are within the entire life sciences and biotechnology ecosystem within the country, and that contribution that you have made to the growth of the ecosystem again is very, very commendable, because you have actually been able to support not just research within your own department, which you have in terms of competitive grants, but contributed to building a very sound and strong biotechnology and life sciences base within the country through the expertise that you have shared, being committee members, being site visit group members, being a part of our strategy planning groups. And I think We do sincerely appreciate that, wish you good luck for this, and we hope that the younger faculty who is coming in also follows on the same footsteps, and we do continuously stay engaged with this particular activity that we have been doing. The DBT, as I said, has been supporting centers of excellence. Originally it was as a separate scheme, but now over the last couple of years, area based centers of excellence, and the Indian Institute of Science itself has a number of such activities in different forms. They may be called centers of excellence, they may be called program support, or they may be called different activities. But what really is the whole basis of these centers of excellence, or the program support activities that we support? While getting excellent, cutting edge science is core to it, and these centers of excellence are built around individuals, built around team leaders. Our hope is that these team leaders would really be the catalysts and be able to attract a large number of younger scientists and researchers to build very strong teams, which has happened in the centers of excellence that we have supported. And I think that again has been a very key contribution which has been made, because creating these centers of excellence is actually identifying those areas of core strength, which then become the central nucleus with which they could collaborate with other groups, either as collaborative projects, or the researchers and students that come out of your centers then move to other institutes and research groups to build similar research excellence quality in other places. So I think that again is a key contribution which this department has made, and we must acknowledge and recognize that, how you have been able to do that. You of course do have the strength, and that I think is definitely a huge appreciation of the Indian Institute of Science and this department in particular, to be able to attract the best brains across the country. I know that for every student to be an alumni of Indian Institute of Science is a matter of prestige and pride and honor, and each one who gets into this system's gates once must be and should feel as a proud and privileged student, because but then there is a huge amount of accountability that is attached to it. If you are an alumni of the Indian Institute of Science, there is a particular role that you need to play for society as you move out of it, and each one of you is groomed to take that as we move ahead. I think today where we stand, and again I quote what Dian Rao said, 100 not out. What is it that you are looking for your next 100 years? I think that's important and critical to see. Beautifully accomplished in these 100 years. But I think one fact we have to all recognize, that the pace with which the movement is moving now is much faster than the pace which was in the last 100 years. So if we have to keep pace with these next 100 years, our speed has to be much faster. We have to be aligned to newer changes which will come in. And this would be not just in terms of the finances, in the modes of funding, in the collaborations, in the various outcome parameters with which departments such as this would be assessed as we move on. And I think that is going to be totally a different scenario from what it was in the last 100 years. And there, I think it's crucially important what you've already embarked upon. Think within the department, how do you more connect better the basic and the translational sciences. And I think that is going to be definitely the future as we move on. The basic sciences, the knowledge generation, the fundamental sciences has to feed into the translational sciences, and today it's no longer a time within our research innovation ecosystem where we say the laboratory has done this research, we've got our research leads, we're still waiting for somebody to come and say, can we transfer it to you and can it move on? I think those days do not exist any longer now. It's all about co-development partnerships. It's all about being a partnership right from the science to the translational part of it. That's really what the ecosystem demands, and that's what's happening all over, and I think that's really what the department has also been able to get into these activities, and that's what we would really like to see this department contributing to. Today we were discussing, and at tea time many members here were asking me about the hike in the DBT budget. Do we have a higher budget? Do we have a planning for the next 5 years? Yes, we do, and Professor Padmanaban spoke about our targets which have been set for us. It's wonderful to have these ambitious targets, but how do we as government even pitch for such ambitious targets? It's based on the confidence that we have on groups like you to be able to deliver that forward. I mean, these targets don't come just from the Department of Biotechnology sitting there. It's based on a large number of conversations and levels of confidence that we get from groups such as this. Yes, India has pitched itself with a huge ambitious target specifically for biotechnology. We have pitched ourselves to say that currently the industry is at a little over 50 billion, and we are hoping we'll close at around 60 this year, but let's see what the figures say at the end of it. So in fact, I must say, sir, that when we pitched the target of 100 billion, when we actually made the target presentations, we were told that we are going too slow on our targets and the target should be 150 billion. So now the department has officially accepted the 150 billion target by 2025, and this is not based on just a statement made by anybody. There are analysis reports which have been brought out, and that was at the Global Bio India. Reports were released by various groups, groups of consultant groups who've done detailed analysis of the sector and said that with the various enablers which are into the ecosystem, India is positioned to be able to achieve 150 billion bioeconomy sector by 2025. Under the Make in India 2.0, as they call it now, what is it that has been positioned? And actually the biomanufacturing has been pitched at about 100 billion now. So the biomanufacturing is going to be all the various biological products. So I think Professor C.N.R. Rao, when he said that he has a lot of hope on the biology side and on the biological sciences, I think he was absolutely correct, because that's where the entire ambitions have been pitched on. And it's all, again, targets that we look towards you to see how we could take that forward, how we could move it ahead. But I think what is also interesting, and this institute clearly exemplifies that, is that it's no longer just for biologists to be able to meet that target of anything related to biology. It's all an interdisciplinary area now. And more as we are moving, it's the interdisciplinary nature of it which is coming out. Any of the programs that we talk about, it's a combination of bringing in the engineering, the clinical research, the computational biology research, the bioinformatics groups, the various groups who get cross-connected to it, a large number of them, who then are being able to respond. Professor C.N.R. Rao spoke about synthetic biology, and Dr. Modak knows how much in DBT we've been trying to work towards, say, can we have strong synthetic biology groups? So I think it's, again, what is it? It's just people with interdisciplinary backgrounds coming together. Can we actually have the bioinformatics group part of this whole system? Can we bring in the computational biology group? Similarly, our whole Genome India initiative, which Professor Padmanaban spoke about, it's the genomic cataloging, but just the cataloging is not going to give us what we want. It has to be the computational part of it, has to be driven towards the application, whether it's terms of the diagnostic and or it's looking at your precision healthcare through therapeutic, they're combination. So that is the initial part of it, but we need to bring in other disciplines. And I think Indian Institute of Science, particularly this department, has a huge role to play as we take it ahead. And that's something that we would really want to encourage you to come forward and say, how do you bring in these connections? How do you look at taking this excellent cutting edge research knowledge that you have towards translation, connecting them and seeing how we take it forward? It's also important that, as I said in terms of the committees, but it's also important to note that each one of you here is a very, very strong mentor. If we talk about these large number of startups, if you just study the profile of these startups, we are very pleased that nearly 50% of them actually are students who come from IITs and other colleges to be able to take on this startup and entrepreneurial venture. Yes, we do stand up and say We do not have the cluster concepts as you have in the US. Why don't we have a Boston cluster? Why don't we have a Cambridge cluster? Or why don't we have a San Diego cluster? I think we still haven't been able to put it into that shape, but at least we have been seeing over the last 4 or 5 years mindsets changing. But these young entrepreneurs, or this young entrepreneurial instinct that we are trying to ignite through the large ecosystem that we are setting up, and Professor Padmanaban spoke about BIRAC, which is one of them, but then you have the DST, which has a very, very strong network. CSIR is setting up its own incubation network. The NEETU Aayog's Atal Innovation Missions network is there. So within the country, there is a very huge network which is accessible to everybody. But what we see is currently the missing gap over there is the strong mentorship which these young students need to be able to get into this entrepreneurial journey. They can get into it, but to be able to succeed in this entrepreneurial journey. So it's all about, our worry today is not about creating a pipeline. I think somewhere we've been able to understand how this pipeline works. Our worry is the scalability and the sustainability of this pipeline. And I think there, mentorships which are provided by departments like yours are key and critical to the scalability and sustainability of this ecosystem. Because we are talking about not just an IT-based startup ecosystem growing. These are solid science-based, technology-based enterprises where cutting-edge science is important for them to take their enterprise forward. And many of you who sit on our committees which actually evaluate these startups, you know that the basic initial evaluation of theirs is done on their science. Once their science is perfect, then we get into their business plans and to their others. So how would they understand that? I think it's all about the mentorship which will come from departments and institutes like yours, because that is what is important and critical for us to move forward. And I would urge you to stay connected with our ecosystem that is being created to see how you can take it forward. Lastly, I would also like to mention 2 more things. And one is that you know that the Department of Biotechnology was the pioneer in building these bioclusters, as we call them. And we have a vibrant biocluster in Bangalore. We have one in the NCR region. One was set up in Kalyani, which was more of the cancer and systemic medicine group, and one at Pune with the NCCS and IISr, which was again more about the deep knowledge, understanding on system biology and those aspects of it. Professor Padmanaban spoke about a recent group which was set up to examine what really is the push that we need to give for the technology sector, and we made these presentations across various ministries. What came out is that our clusters so far are research institutes working with startups. If we have to complete this whole circle, we need and we cannot leave the university on one end of it, which is again a very crucial and important component which actually provides the knowledge base, the deep knowledge base, and the industry on the other side, the large industry on the other side. So we've actually been able to get approval to set up what we call knowledge translational clusters. And if those of you who followed the finance minister's budget speech would have actually seen that mentioned as one of the recommendations, that we will set up knowledge translational clusters across different fields. And definitely the biotechnology field, we've already got our approvals more or less in place, and we hope in the next 2 to 3 years these 4 clusters, including these 4 clusters, we'll have at least about 10 such clusters in the country, which will bring together the universities, the research institutes, the startups, and more importantly the industry, to be co-located for co-developmental partnership programs. And for this, I would see Indian Institute of Science playing a very major role, not just because you have the ingredients of what this cluster can actually combine. The Bangalore cluster exists, becoming part of it, but I think the strengths that you have, and that is what we would want to see in this cluster, is reaching out to a larger expanse of students, reaching out to the tier 2 and the tier 3 cities, where we know that there is a lot of interest, there is enough of talent, and there is an inquisitive mind sitting there. It's all about bringing them into the fold of this, and it's about giving them this culture this cultural ecosystem so that they could also thrive. And we would definitely like to see this department playing a very major role into it. Many of our schemes, whether it's Builder or Sehaj, is all about giving access to universities, giving access to the larger facilities on the shared infrastructure. But I think this really is what we would want this department to play a major role in being able to build not just the expertise or the competence levels within this, but make them deeper foundations, but also expand so that we have a larger critical mass of students. Professor C.N. Rao very rightly said, for each name can we think of 5 to come in? How is that going to happen? We have to spread, we have to bring into the fold a larger number of institutes, and I think that really is what will give us our achieved target of moving whatever steps we are. We spoke about India being in the publications. Yes, we may be third as it has been pointed out, but we are way behind in the third. So I think the first and second are much ahead of us. Is there some way we can catch up to that second position? The gap is huge. But I think we do have the potential, and that potential comes again from the lot of exciting research work which has been done. So it's not just the biotechnology, but the whole research and science ecosystem where we see a lot of potential which exists within this department, and these young students who are here, who are our future leaders, who are our future researchers, to take it forward. So with this, I would just once again like to congratulate each one of you for being part of this 100th year, and I'm sure many of these students here are going to be part of the journey for the next 50, and then many more to come in for the next 100 as we move on. But this is the foundation which is going to be important and crucial for the country to be where it is as it moves on. So congratulations once again, and thank you very much for inviting me to be a part of your 100th year celebration. Thank you.
Thank you so much, ma'am, for this wonderful talk. And again, I'd invite Professor Padmanaban to please present her with a token of our appreciation. Next lecture, the next speaker is Dr. MAHATAB, BAMJI, and I'd request Professor Padmanaban to please introduce the speaker.
Passionate lecture. The message is clear. Good science is okay, publications are okay, but we need translation. The society needs science to be applied, at least part of it. I can't expect every paper will become a product, but that's the message she has given. Thank you, ma'am, for all the effort you have taken to come. She reached Bangalore at 1:30 or 1:30 a.m. or something like that. I honestly did not know I have to introduce Dr. BAMJI and Dr. KRISHNASWAMY. So I have a write-up, but MAHATAB, as we all know her, is still a live wire. We know her running between that building and biochemistry building, as she was with Professor KUMAR. Outspoken, I don't know how to describe her, but before that, let me read what is written here. Retired director grade scientist from the National Institute of Nutrition and currently an INSA honorary scientist. She has contributed significantly in the field of nutritional biochemistry, specifically developing biochemical tests for early detection of some B vitamin deficiencies and derived their requirement, contributing philanthropy with her scientific endeavors. She joined Dangoria Charitable Trust, and this is her real inspiration, this charitable trust. I'm sure she's going to mention something about what this NGO is doing. ICMR's emeritus medical scientist. She has been a recipient of several notable awards and honors, some of which include National Award for Women Scientists 2000, C.V. Raman Medal of INSA 2005, and the list goes on. She is hailed as a living legend by the International Union of Nutrition Sciences. We all know her deep interest in vitamin A, and really I find her trust now. She will talk about that, doing enormous contribution to the society. Dr. MAHATAB, BAMJI, please.
RENU, you have made women scientists proud by breaking, as it were, the glass ceiling. We need more RENUs in this country. I'm so happy to be here. It's an emotional moment for me. The reason I accepted to come here was not just to listen to science, but even more, I'm afraid I have to say, to meet my old colleagues. And for that, I really thank the organizers. I wish this department continuing success, and I hope it reaches greater heights, so that it will be one of the best institutions, better than any in China. Now, you see, it used to be said that it's difficult to get into the institute, more difficult to get on, and most difficult to get out. But fortunately for me, none of this was true. I got into the institute without even dreaming about it. Professor Giri was the examiner for MSc. And after the Viva Voce, he said, would you like to come to Bangalore and do your PhD? Dr. Kama is looking for students. So without thinking, I said, oh, yes. When I went home, there was a little, my mother and my aunt said, how can a young girl go alone from Bombay to Bangalore, an unknown place? Impossible. Then, of course, my father convinced them, and I joined the institute in 1957. That was before most of you, except some of you. If you ask me to describe my stay in the institute, I can say it in 2 words: stressfully happy. Anybody who has done PhD research knows that it is a stressful experience. Today, in those days, we had plenty of water and electricity, but no equipment. Today, you have a lot of equipment, but no water and electricity. And it was happy because I had wonderful colleagues. I still remember Vaidyanathan, late Vaidyanathan, sitting across the table and showing me paper chromatography. Mahadevan, S.A. Kumar. But Padmanaban came later, after. He's junior. So, and in the institute, I developed my love for Carnatic music. I was always very fond of Hindustani music, and I developed my fondness for reading Hindu paper. Some of my friends tell me now that your mind is perverted because you are reading Hindu. It was very nice to work with Professor Kama. I had a very wonderful colleague in P.R. Sundaresan. And the project that was given to us was studying the metabolism of vitamin A2. Now, vitamin A everybody knows, but there is a thing called vitamin A2, which occurs in some freshwater fish like Valaga atu. And we had to extract that from Valaga atu, and that required petroleum ether. But pure petroleum ether was not available in those days. So in Dr. Kama's lab, there were always distillation going on for petroleum ether. Then we had to run column chromatography and then identify the samples, and that had to be done on a Beckman DU. Prize instrument, only one between biochemistry, pharmacology, and microbiology. So we had to register our names. And my name invariably came, I preferred it after dinner. So I was the first one in the mess to go at 7:30 and run to do Then Professor Kama will come in the night. He used to go every day to the club to play bridge. Here we were struggling away and he was playing bridge. So he would come, and then of course he would bring chocolates and sweets and say, what's going on? We had to crystallize vitamin A2, for which we required carbon dioxide, solid carbon dioxide. How did we get it? It was not available. So we had to open a cylinder of carbon dioxide, put a flannel bag to it, and then pat, 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 and get some fused crystals of dry ice. So one day we succeeded in crystallizing vitamin A2. It was around 12:31 in the night, and we took a photograph also, and put it in dry ice. But next day I came and I found it had got oxidized. There it was. Anyway, so it was, but in those days, I think it's so even now, institute has a culture of working round the clock. So there was nobody asked you when you came, when you went, what you did. But there were those who did not come, did not do anything, and they were the ones who were difficult to get out type. So then in one day, Professor Kama came with Sir C.V. Raman. I was, oh, C.V. Raman. So Raman had a new theory for dark adaptation. See, the conventional theory is that when you enter from a bright place to a dark place, pigment rhodopsin forms in the retina, which is a combination of opsin and vitamin A aldehyde. And when you come out, so that's why it takes a little time, dark adaptation. And when you come out, rhodopsin breaks. Raman felt that this will result in a lot of loss of energy, so this cannot work. So he said, the active principle is lutein, which is in the retina. And this gave me an idea of how a brilliant mind works. He used to sit across the table, drag me to the library. I would take out all the papers of George Wald and read. Anyway, he gave a good seminar also. Unfortunately, that theory was not accepted and it died a natural death. Then another reason why my life was happy here, there, we had a nice hostel. We were only 20 of us. So all men were many. So they kept good track of us. They knew everything about us. We didn't know anything. So with the result, I didn't know that some people who are eminent scientists were at that time in the institute. And later I came to know that they were in the institute. They knew about me, I didn't know about them. The mess was very nice. There were only one vegetarian mess, one non-vegetarian mess. I was in the non-vegetarian mess. And there were two very devoted, caring servers, Raman and Chandra. So they knew who wanted chapati with ghee, who wanted chapati dry. And so for some reason, I had developed this taste for getting fried egg laid on idli and served. And Chandra remembered it. Many years later, I came to the institute and stayed in the guest house. Chandra saw me, I saw him. We were both delighted to meet each other. And then with everybody sitting for breakfast, Chandra came and said, Madam, idli today. So I said, oh, very good. Fried egg sitting on idli? Then he told everybody that Madam likes fried egg to be sitting on the idli and served. So that was that. Then Professor Sharma came. Professor Giri died. He was a wonderful person, very gentle, a true scientist. He died in 1958, only 1 year after I joined. And Professor Sharma came. Sharma was a good scientist, a disciplinarian. And he brought in the system of journal club, where all of us, for every journal club, all of us had to go prepared with a paper. And in those days, no PowerPoint, no slides, blackboard and chalk. So we had to go, and we were all very nervous. One day I was called, and I still remember I presented a paper of Dr. Patwardhan, on mechanism of action of vitamin D. And Sharma liked my presentation and gave a faint smile that made my day. After that, Sharma he started many new things. And after leaving the institute, I went to Boston to Tufts University, then to Johns Hopkins. And then when I wanted to come back, I wrote to Sharma that please help me to get a placement. So he said, write to Dr. Gopalan in National Institute of Nutrition, which I did. And I got a job in NIN, which I took in 1965 and retired in 1994. So Giri helped me to come to the institute. Sharma helped me to go get a job in NIN. And I'm ever so grateful to these two great men. And Kama helped me to get PhD. Now, I have given a write-up for the book that is being brought out. So I wrote to DN that since I'm giving this write-up, I will speak about something about nutrition, flag nutrition problem. He said, it's okay. Then I said, when is the book coming out? He said, it will take time. I said, then in that case, I might as well share my experiences in the institute. But yesterday they said, you also speak about some of the work that you're doing in nutrition. You see, having worked in National Institute for Nutrition for so many years, you learn something. And somehow I always wanted to get out of lab and go into field work. I was not qualified for it. I was not a medical person. I was not an agriculture scientist. But I wanted to do it. Fortunately, at that time, my colleague, my friend, Dr. Devyani Dangoria, had started a hospital in 1969. She started that hospital in a village called Narsapur in Medak district. It was in the middle of forest, lovely place. 94, the Dangoria Charitable Trust started a home for the aged. Please don't think that in India, old people are well taken care of by the children. That's a myth. If you are well taken care of as in your old age, you are lucky. Not everybody is so lucky. So you can come and meet our old people and you can write a multi-story book on that. Anyway, so Devyani said, why don't you join here? Fortunately, at that time, Department of Science and Technology started a program, Science and Society program. And later, DBT also started. So now the question was, nutrition. You see, nutrition security is not just food security. Nutrition security goes beyond food security. And for nutrition security, you have to have safe drinking water, environment, healthcare outreach for prompt treatment of infections, etc. Now, after over 70 years of independence, today we don't see the frank deficiency diseases like blindness due to vitamin A deficiency or pellagra or scurvy or any of those. But subclinical deficiency is very much there, and anemia is #1 problem. I hope all of you know what is development goals and UN development goals, national development goals. There are 17 of them. In that, the first goal is zero hunger. Fifth goal is gender equity. Then there is education, sanitation, etc., etc. And NEETU Aayog every year, these goals have to be reached by, I think, 25. And NEETU Aayog every year does an assessment of to what extent different states have reached that goal. And you see, I didn't bring the slide. The worst performer is hunger index. And hunger index is not just food, it's also nutrition, it's farmers' income, etc., etc. But this is not to say India doesn't have enough food. Today we don't have place to store food. Yes, we were very short on pulses, but in last 2 years, even pulse production has come up. India is #1 in horticulture production, number 1 or 2 in milk production. And even then, why is there hunger? One of the reasons is the farmer who produces finds it too expensive to eat, he prefers to sell it. So the challenge is to produce it at lower cost. And equity, and another area where we are performing very badly is gender equity. One of the worst performers. And equity, even economic equity. Ahmedabad, Gujarat, which has done very well in economic development, if you see their poverty index, it's still very poor. Anyway, so we identified 2 areas where we needed to work. Now, if you look at Indian diets, which are cereal-based, if you have enough cereal, you will have enough protein, though protein quality will be a problem. But what Indian diet lacks is micronutrients, vitamins and minerals. It's very deficient. Vitamin A, riboflavin, iron, and so on. Now, there is no need for vitamin A deficiency to be there because in the nature, there is enough beta-carotene, and as you may know, beta-carotene is a precursor of vitamin A. So it's a question of not knowing what to grow. So we said, then the other issue that we thought we will take up is complementary feeding. According to WHO guideline, breastfeeding should start within 1 hour of birth. Whereas in our area in those days, first 3 days of milk used to be discarded, chedipal, bad milk. Colostrum used to be discarded. And yet colostrum is one of the richest sources of nutrients. So this was one issue which doesn't cost money to teach people to start. And I'm happy to say that today, at least in our area, that parameter, starting breastfeeding from first hour, that has caught up. The second one is, as I said, micronutrient deficiency. And we said we'll concentrate on what I called nutritionally sensitive and environmentally sustainable agriculture. In our area, they go mostly, it's a water-stressed area, and still they grow paddy and sugarcane, most water-demanding crops. And most of our farmers are small and marginal, 2 acres, 3 acres. The average is about 2.5 acres land. To make a farmer change his cropping pattern is extremely difficult. But we had to work with them and say that keep aside 2 guntas, 40 guntas make 1 acre, 2 guntas, 3 guntas for growing vegetables, but not lauki and turai, not gourds, which are low, but micronutrient-rich vegetables. And amongst them, green leafy vegetables are the treasure trove. So we are emphasizing on green leafy vegetables, beans, because beans also enrich the soil. So we are trying to convince the farmers to grow and also it's not enough to convince the farmers. You have to change the mindset of agriculture extension workers. For them, income and export is the main priority. So this is where we are working now. And for complementary feeding, the WHO guidelines are breast milk alone till 6 months. And after that, complementary feeding. But our women, we don't have to tell them about breastfeeding. Maybe urban women don't do, but rural women do. But they don't give complementary food till 1 year. So, complementary feeding. Then I found that many pediatricians would write Ferrex, Cerelac, and the mothers cannot afford, so she would put water and dilute it. So we developed a low-cost complementary food, which we have labeled as Poshana. It is nothing but it is suji, chana dal, groundnut fortified with iron. And today that product is being bought by a number of hospitals, Ramakrishna Math Health Center, etc. Then one fine day, my neighbor came and said, "Auntie, do you prepare uggu?" I said, "For God's sake, what is uggu?" So she said, "Uggu nahi malum aapko?" I said, "Nahi malum." Uggu is the best complementary food you can think of, and that the people are preparing. It's a combination of rice with minagapappu, the rice, the combination of dals. And I never knew that villagers prepare that. You see, we make effort to find out what they know and try to, but this we had never done. So we said, look, this is another uggu. Now, village women are busy now. They don't have time to roast, powder, all that. So if they have something cheap, inexpensive. But now, fortunately, in Telangana, I think in Andhra Pradesh also, ICDS is supplying such a ready-to-cook food. Another time when I found something, we asked them, what causes diarrhea? They would say, enda tagalte, sunstroke. Some of them will say, if you eat indigestion, etc. But they don't link diarrhea with bacteria. What causes malaria? Domalu kuttiste. They immediately link malaria with mosquitoes. Then what do you do when you have diarrhea? We go to the doctor. Then then one woman said, we give sabudana. Excellent. So while working I realized that working in the field, don't think they don't know anything. First try to find out what they know. That is the mistake government is making. Making plans in air-conditioned room, taking the scheme to the people. Now iron folic acid tablet for anemia. It's a good program, but people they throw away the tablets. Why? Because they get black motions. So they have to be educated. So as I said, for nutrition security, you need 3 A's: awareness and access at affordable costs, to balanced diet and right feeding practices when it comes to children. Safe drinking water is the most important thing. And now I'm happy to tell you that one scheme of the government that has worked is Swachh Bharat. Today in our area, every household has latrine. And water connections are also there. And healthcare outreach. So over the years, I don't think I have much time. So please, we have a website, www.damgoriatrust.org.in. So we are trying to prepare models, strategies for each of these. And since we try to assess the impact, and impact assessment is done through finding out how many gardens. Yes, we are using organic methods like vermicompost, botanical pesticides, etc. So impact assessment is done by finding out the acceptance of technology, but technology without behavioral change communication is hopeless. Highest priority has to be given in awareness generation. And that's what we are doing now. And over the years, we have received support from DSC and DBT, but now DBT doesn't give support, doesn't allow people above 70 to be principal investigators. DSC does. So now we have set up a hub for food and nutrition security. So we take 5 villages every 6 months. And another very useful introduction that we have made is poultry. High egg yielding birds. These birds lay 140, 50 eggs per year against country birds which are 35, 40. And now we have been able to do all this because of the support I've received from agriculture scientists, veterinary scientists, medical scientists, and all that. So now every 6 months we take up different villages. So now our mission at 85, now my mission is to go as far as possible and create nutrition security in as many villages as possible, and we are trying to get corporate support. Thank you.
Thank you so much, Dr. BAMJI. It was a very wonderful talk. The next speaker is Dr. KRISHNASWAMY. I request Professor Padmanaban to please introduce him.
Thank you, MAHATAB, for a great lecture. Still can realize her passion, her commitment. Your junior, Padmanaban, is 82 years old, MAHATAB. catching up very fast. I have great pleasure in introducing Dr. P.R. Krishnaswamy. PRK, as he is fondly called, is the oldest living alumnus of the department. Still going strong, but first let me read what is given to me. Founding faculty and chief of pathology of the pathology laboratories at Jaslok Hospital and Research Center in Mumbai, holds one of the patents for recombinant insulin in India, an alumnus of the Department of Biochemistry. His work on the mechanism of glutamine synthetase using the ingenious chase method to trap isotopically labeled intermediates is well recognized and used for several other enzyme mechanistic studies. His contribution to unravel the metabolism of 5-oxoproline have led to the discovery of 5-oxoprolinase, an enzyme whose clinical importance is currently a subject of several studies. His creditable accomplishments include outcomes of study on post-translational glycation of proteins in diabetes, notably hemoglobin, HbA1c, early introduction of it as a clinical chemical test by his laboratory at Jaslok Hospital in the mid-1970s. You know, he's still very active. He's associated with our nanoscience department at the institute. We regularly meet, if not regularly, once in a way we meet and then exchange notes. He knows so much about hospital administration. He knows so much about medical issues of facing the country. And when he gets exasperated with some of the things that are happening there that should not be happening. So, I can go on and on about his passion. Just like MAHATAB, he has several interests. This can be done, that can be done. And in nanoscience, he is very much interested in converting many of the tests in a nano form. And his advice is very valuable, and I look forward to meeting him and talking to him. We are anxious to hear you, PRK, your experiences. Yeah.
Friends, I think it must be getting impatient for lunch. I will not hold you too long because unlike MAHATAB, whose journey from her post-graduation to Bangalore, later on to a career, was in straight lines. Bombay to Bangalore, Bangalore to Boston, Boston back to Hyderabad, Boston and somewhere else to Hyderabad. My journey along my professional path has been meandering. I don't know how many of you really know the dictionary meaning of the word meandering. Meandering can also dry up at one end, but in the case of the rivers, good rivers, it doesn't end till it gets into the ocean. Maybe the God Almighty might have blessed me to see the ocean someday, so I think it is still running, but very, very slowly. I thought I'll take this opportunity today to pay my tribute to my mentors, whose influence really facilitated and illuminated the meandering journey in which I also engaged in some reasonable biochemistry. Because I was fortunate during my work in the institute and certainly during my postdoctoral work with Meister, the late Dr. Meister, to accomplish something in basic biochemistry, particularly enzymology, which had set me on fire. But then on return to this country, like it happened to many people, there were no seats available. So I had to really make a number of detours, work in several places. But wherever I did, as I will describe to you as we go along, I think some contributions were possible. The two mentors in this institute to whom I owe my entire career and whatever I have done are the late Professor Srinivasaya and the other, Professor Giri, to whom I switched upon the retirement of Professor Srinivasaya to work, who Giri very gladly accepted me and Dr. Kalyankar. Two of us chose to work with Dr. Srinivasaya. I will call him a doctor, super doctor, because I think his reputation as a igniter of minds, as a super guide, as a very versatile genius, if you want to call him, at that age of retirement of 55, was known throughout the country, in fact throughout the world. So I may spend a little time on talking about him, and later a little bit on Professor Giri, who so willingly accepted us, because both of us brought the research problems that we were getting interested in working with Srinivasaya, Professor Srinivasaya. And Professor Giri was extremely generous, because I think he had not worked on microorganisms. I chose to work on riboflavin excreting mutant yeast, which one of the illustrious cytogeneticists in this institute, Professor M.K. Subramaniam, had prepared. He had produced it, and he had published extensively. I think so many of the Nature papers you see there are his. And at the time he published those papers, it created such a controversy. It was really very, very difficult for him, in fact embarrassing for him, because many people thought it was not really reliable work. But it took decades later that he was proven so correct. But I got interested in one of those mutant yeasts, and I wanted to do some good biochemistry on them in relation to the biosynthesis of riboflavin, which it was secreting, and also to work on what was very dear to Dr. Giri, chromatography. Giri was a simple person, but profound in terms of his deep interest in science. I think I say this in all seriousness, because I think MAHATAB said Kama used to be in the club when she was in the lab. I think with Professor Giri, humorously put, it was the other way around. If you set up a chromatogram in the lab in the evening, and when he was going out home, he got an indication you will go home and come back to remove the chromatogram after it is run. He would say, you don't bother to come. I will come after dinner and remove it. I think he was engaged in work, and next morning he would never forget to ask me what that chromatogram looked like. I think he was a teacher of that kind. So for me, I think the experiences with both of them was extraordinarily pleasant. Mentorship in modern science has a, I think I have to use this to take to the next slide. This one? This?
Yes.
Mentorship in modern science has a very short memory of those who contributed to it. This is said by Arthur Kornberg very recently in one of the JBC issues, where he writes about his mentors. His mentors were Corys, Gertie and Teresa Corey and Ochoa, and he very neatly describes this. Maybe it will get perhaps even shorter. But I think in this institution, obviously it is not going to, because I think when I saw very briefly this morning the manner in which the student community, which is organizing this, and some faculty, have really unearthed so much material about the past. I'm sure there are treasures even more waiting for you to unearth. If I can be of any help in that, I will dig with you. I think we can find. I think this is a remarkable thing. I think this is a very important thing for science. Memories of who contributed and how, because more than the contributions, the process is the most inspiring of all things, number one. The second is memories are not just only for information. Actually, a good mentor is one who ignites a fire, kindles a fire, that it lasts in you forever till your end. I think that is, a mentor. I think very hard to get mentors of that kind. Because mostly, I think we get filled with information, filled with techniques, filled with ways of doing things. That is not quite mentorship. Mentorship, we should not use the mind as a vessel in which you throw a lot of information and techniques and processes. I think your mind has to be set on fire. Professor Srinivasaya was a legend. He was indeed a legend. He essentially was an intellectual, very impatient of shoddy work, lack of originality, and most important of all, vagueness in expression. If somebody was vague, I think in scientific communication, he had very little patience for it. Because I think he taught you, though he was impatient, he would teach you. He would scold you first, but then teach you the art of expression. And I think his intellectual prowesses were extraordinary. In fact, before joining, I did some research by talking to people who work with him, looking at the literature. And for those of us in the Central College, in those days, current science was familiar, because current science, which he began, he used to mimeograph, that is cyclostyle. The first issues of current science were cyclostyle. He used to do it himself. He was also an institution builder. In fact, he was one of the members who started the Society of Biological Chemists. He had a tremendous organizing capacity. He was very fond of music. In fact, I given 2 photographs, one when he was young. I had not seen him at the time. The 1952 is the picture that I saw him. And also, in the same slide, I indicated his specialities were biochemistry, microbiology, plant virology, enzymology, biotechnology, in many ways, and instrumentation. He was good at all that. Many of the equipment which he described in his papers, glass equipment, gadgets, he would fabricate himself. He was a very good glass blower. Dr. Kalyankar and I, who were his last 2 students, we used to get lessons from him, lectures in his office, starting at 8:30 in the morning. 7:30 he would come. 7:30 to 8 o'clock, he would show us glass blowing. In fact, I think that skill of glass blowing still remains with me, because we used to make our own pipettes, pasture pipettes, many other gadgets, calibrations, all this he would teach us. Meticulous, absolutely meticulous. Then he would lecture us on a variety of topics, and he had the dilatometry, for which he was famous, in one corner of the lab, and do some experiments for us. This is the famous dilatometric study work on enzyme action that he and Shastri, who went to become the very big, well-recognized scientist in the CSIR, he wrote the Wealth of India volumes. They are monumental volumes on the natural wealth of this country. This is their paper. Then you can see the paper was published in 31 in Nature, and he was a very frequent contributor to Nature. Most of the work he did in dilatometry and many other things, he would publish only in Nature or Biochemical Journal or in the Journal of Biological Chemistry. And then when the Biochemica Biophysica Acta started, you will be astonished, among the scientific advisory boards drawn from the globe, apart from the editors from where they were published elsewhere, Srinivasaya's name was there, along with Sir Hans Krebs, Euler, some of the greatest authorities. You should also remember, till then, he had not made any trip abroad. He was known only from this country. In fact, any paper he wrote to any of these journals was accepted in total, without any correction, without any modification, without any questions. Not only that, there is a very beautiful paper You should get that little piece of paper. He wrote a paper on mannitol in sandal leaves. I think in one condition, the sandal leaves produce mannitol. And he made this observation and characterized mannitol. He was a very, very good chemist. He was a biologist whose chemistry was absolutely of very high standards. He characterized all of that and wrote a 6-sentence note in ink. His handwriting used to be perfect. Those lines he wrote, and that paper was sent to Nature in the handwritten form. And Nature accepted and printed that. In fact, this happened to me during the last year. Dr. Giri asked me to send a paper on one of the things that we did with Giri, and I didn't have the time. The typewriter, there was only one typewriter in the department. That was out of order. So I hand wrote, and in the old tradition, I sent it to Nature. And to my surprise, I found after a year, the paper had appeared in print without any change. This is in the utilization of 4-ACA, 4-aminoimidazole carboxamide. That was the tradition. I think we should thank Srinivasaya for that. They thought anything coming from this department must be so good. The other, I'm just giving for the benefit of the younger people here, what a pride we can take in some of these people who built this department. Lindström Lang was a legendary figure in my generation and also in the the future generations for protein chemistry. His lab, Carlsberg Laboratory, which was maintained by a brewery industry in that country, was a lab where it was like a mecca for people who wanted to learn protein physical chemistry, protein structure, protein function, enzymology. The big names like Sheraga, Richards, many American protein chemists you hear of the 50s and 60s were all frequent fellows visiting his lab and working there. In fact, he refers in a paper in Nature, both in the beginning and in the end, in total agreement with Srinivasaya and his colleagues' findings in the year 1937. But he modified, improved, and refined that instrument. But the credit for dilatometry and similar measurements and enzyme activity entirely belongs to Dr. Srinivasaya. And his interest in microbiology extended to the level that his lab became the center for collection of lactobacilli, which had special requirements, individual requirement for amino acids. See, in those days, estimating individual amino acids were a very big problem. You had to have chemical methods, very tedious and difficult. So a microbiological methods by which each lactobacilli required a particular essential amino acid would be utilized for assay of amino acids. In fact, he taught me and made me do assays for all amino acids, all essential amino acids using lactobacilli. He had the national collection of type cultures. In fact, Kalyankar and I were given the task of studying the entire nitrogen balance in the life cycle of the silkworm by starting with about 500 eggs, from the egg stage to the pupal stage. We had to follow up the food intake, the amount of silk produced, weight, body weight, and nitrogen. Then we made ourselves about 200 Keldal flasks, small Keldal flasks, because we knew glass blowing, set up a factory to do nitrogen estimation manually, and we submitted this report to Mr. Srinivasaya. Poor man, at that time, I think he retired, but then he kept in touch with us. Giri essentially was a very, very soft-hearted, childlike person whose curiosity in science absolutely was infectious. I think when he got enthusiastic about something, I think he would set everybody on fire by his smile, by his calling a person by the wrong name. Appaji Rao has suffered it. Appaji Rao is here. Appaji Rao used to suffer. He used to call him Kupurao or something or the other. But then Appurao, when he joined, I think we collaborated together. We did a lot of work on flavokinase. And I requested Dr. Giri to permit me to work on these microorganisms and devote my time to studying the riboflavin synthesis, biosynthesis, and also the enzymes of FAD and FMN making enzymes in the yeast systems. And he was very generous to permit me. And I want to emphasize this, I will take this occasion to say that there was a certain sense of skepticism and almost very painful ridicule. The purists, the orthodox people used to really attempt and do about Dr. Giri being engaged in chromatography as his main passion. How wrong they were. See, at that time, in the history of biochemistry, separation techniques were the greatest need for biomolecule separation and study. And circular chromatography, by virtue of its uniqueness of speed, separation, and most importantly, quantitative extraction from the bands that you identify. They may be amino acids, they may be purines, they may be anything. That was very easy in those days. So if you look at the wide applications that he employed it for, you will be astonished. Flavins are one example, and we should not forget to mention Dr. Radhakrishnan's finding of allo-hydroxyproline was in circular chromatography. Many things happened like this, I think, in the department. Amino acids, carbohydrates, and also I did a piece of work with him along with Professor P.L.N. Rao, where we separated the degradation products of purines and nucleic acids, among one of which was the precursor for adenine, purines. We separated. We could detect it only because of the sensitivity and certain features of circular paper chromatography. And the application to flavin was very well appreciated because I think these were very, very expensive nucleotides in those days. I think if you made it biosynthetically, enzymatically synthesized, one could isolate them. In fact, both Dr. Radhakrishnan and myself, when we did our postdoctoral work in the United States, very successfully applied circular chromatography to prepare pure glutamic acid. If in those days, if I bought from Nuclear Chicago 5 millicuries of, let us say, L-glutamic acid, and I want to use it in a reaction in pure form, if I ran a circular chromatogram, I would get at least 25 concentric circles, radioactive. That means it had 24 impurities. One band was glutamic acid. The rest in different quantities, because its radioactivity, it would show up. So I very successfully employed paper chromatography whenever an isotopic substrate or a compound had to be purified. That proved to be the best method. I used it for glutamic acid, I used it for nucleotide, I used it for P32, for example. I think that method had application in the context of what was the need in those days. These are the papers I published with Dr. Giri. Now I will also take this occasion to mention two other mentors, though they were not IISc faculty, but they had appreciation for what IISc produced. One was the person with whom I did the postdoctoral fellowship. Upon his writing to Professor Giri, he must have seen a couple of our papers, and also he had the occasion to have Dr. Radhakrishnan, who preceded me to him. He must have had an idea of the quality of work that is done in this institution. So he wrote to Professor Giri whether I can be spared for a few years because he was starting a program on protein synthesis. He was a very young man. He became a chairman of the Department of Biochemistry at the Tufts New England Medical Center at the age of 33. He was an MD. He did his residency in the neurology. Then he hung his diploma in his closet. For any visitor, he would say that he was a biochemist, but his diploma of in medicine, MD degree, is in the closet. I worked with him for several years on amino acid activation, its properties, and its reversibility, the ATP synthesis and all the rest of it. But one piece of work which I did with him became a classic because he was quite interested in the synthesis of glutamine from glutamic acid. and the mechanism. The mechanistic explanation of glutamine synthetase as a stepwise reaction was published in the year 1960, and a detailed paper followed the same year. Simply put, the mechanism involved a stepwise binding of glutamic acid and ATP to form a highly reactive anhydride, gamma anhydride of glutamic acid, which was bound tightly, which would react with ammonia, then form glutamine and get released. There was no evidence for this. They all thought it was a concerted reaction. But my work proved the point that there is a stepwise binding, and you form oxoproline if you really heat that only glutamic acid and C14 glutamic acid, C14 glutamic acid and ATP molecules. So this became very important in the mechanistic explanations for many reactions, including the reaction which of ubiquitination of rose. For 60 years later, mind you, I think it is 60 years later after this work was published, this paper was cited by Daniel Purich in 2010 about the enzyme catalysis and control in the book, that this mechanism proposed is a universal mechanism for carboxyl activation. Irving Rose, who got the Nobel Prize for ubiquitination, essentially used this principle, and that activation was understood in mechanistic terms. I'm not going to into the detail. So my interest in oxoproline was remaining still. So I think I had a small fellowship in Mysore, and my good colleague Rajgopal Rao, an alumnus of this institute, a brilliant biochemist, unfortunately is very ill, otherwise I don't think he would have been a great enthusiastic participant in this meeting, very good in metabolic work and amino acids. We together showed for the first time this oxoproline is a very active compound because it gets oxidized in the human to glutamic acid by using ATP. This was not known. For almost 200 years, oxoproline was an ignored compound. You will be surprised. For 200 years, though this compound was known, its metabolic role or its place in the amino acid metabolic events was not defined. But this was done in Mysore by doing a very simple experiment. In fact, we made this glassware, Gopal and myself in Mysore. My glass blowing came in handy. With a 200-time rat, we injected 5 microcuries of glutamic acid, and we got very high oxidation. From Mysore, I had to move. I think the reason being, after this, I became jobless. CSIR had no mechanism to absorb. CSIR didn't have jobs, and the doors were shut here. So I went to Bombay to a hospital which was about to be set in the private sector. This was the Jaslok Hospital. Dr. Shantilal Mehta, who was a founder, was a former dean of the Grand Medical College. I knew him well through some pediatricians because of my clinical interests. And he had a conviction that this new hospital which is going to be set, which is very modern, should have a culture of clinical investigations, clinical chemical investigations and pathology headed by a scientist rather than an MD pathologist, because I think things were changing. They had pathologists working in the lab for pathology, but the entire lab's planning and management was given to me. So the one highlight of that place was I was able to rekindle my interest in glycation of proteins, hemoglobin. I was able to develop a method for glycated hemoglobin, a chemical method. This hospital introduced this for the first time as a monitoring test for diabetes. Jaslok was the first hospital to do it in India. And I think, in fact, in the world scenario, in the diabetic field, India was one of the early countries where a chemical method was used for monitoring diabetes. Later on, many methods. These methods were using ion exchange chromatography. They used isoelectric focusing and so forth. They're more expensive, but they proved to be less specific. So my journey, when I came to Bangalore, ended in the institute through a very interesting history. I met Professor Balram to share my interest in glycated hemoglobin, and he evinced interest in the same topic. And then wherever there are artifactual increases of hemoglobin A1C, where they were beta thalassemix, those cases we were able to prove that these were artifacts, and we found a number of new mutations, hemoglobinopathies. And he introduced me to Professor Nokant Bhatt, who was looking for some assistance or participation in doing a little work on glycated hemoglobin for biosensors. And since then, life has come one full circle, and I've thoroughly enjoyed working in the nanosciences, because this has a possibility of really extending to a lot of other things. Today, for example, we have a very good understanding of glycation of albumin as a short-term marker. Not only short-term marker, but it has a lot of pathoetiological significance, because albumin, unlike hemoglobin, is a conveyor of a lot of items to various parts of the cell. It is also an antioxidant, so glycation of albumin has some pathoetiological significance. Similarly, many other proteins can be looked at. So all this is possible, my dear friends, in my life till now, because I think something was kindled in me when I joined this place by two mentors, Srinivasaya and Giri, and it continues to march on, sitting, not standing. Thank you.
Thank you, Dr. Krishnaswamy. Now, the vote of thanks will be delivered by Big Secretary, Ms. Sohini Guha.
Take a couple of minutes. Distinguished guests, it is my privilege to propose a vote of thanks for this occasion. On behalf of the Biochemical Society, the students, faculty, and the staff of biochemistry, I would like to thank Professor C.N.R. Rao for delivering the inaugural address, Dr. RENU SWAROOP, Secretary DBT, for delivering the keynote address, as well as for inaugurating the exhibition shortly after this, Professor Anurag Kumar, Director IISc, for his time and for accepting our invitation to grace this event. I thank Professor M.R.S. Rao and Professor G. Padmanaban for chairing the previous sessions. I would also like to thank Professor P.R. Krishnaswamy. As Professor G.P. mentioned, he's our oldest alumnus, as well as Dr. MAHATAB BAMJI for coming from Hyderabad to attend this event. I would like to thank all the guests who are scheduled to speak in the afternoon sessions today. I would also like to thank all the alumni who have graced this event, as well as those who have conveyed their good wishes through emails. I thank Professor Varshne, our divisional chair, for his cooperation, as well as for providing the auditorium to conduct this event. Next, I'd like to thank all the faculty of the Department of Biochemistry, both past and present. Professor D.N. Rao, Chair of the Centenary Celebrations Committee, Professor RANGARAJAN, Chair of the Department of Biochemistry, all the students of biochemistry, thank you everyone for your efforts in organizing this event. I'd like to thank all the biochemistry staff members, Mr. Mohan, the auditorium staff, Mr. Achar, Mr. Shiv Kumar, for all their help. A big thanks to the entire exhibition committee, comprising both students and faculties, the archives publications cell for their untiring efforts. Special thanks to Current Science for publishing an editorial titled 100 Years of Biochemistry at the Indian Institute of Science, which was written by Professor Rangarajan, and for featuring the sketch of the old biochemistry building as their cover page this month. Lastly, I would like to thank all the alumni for their generous contributions, DBT, IISc, IIFPT Thanjavur, as well as our corporate sponsors, Indian Immunologicals, as well as Clever Gene. And if I have missed out on anyone, I apologize. We thank you, one and all. Thank you.